Good morning to everyone, and firstly, I'd like to thank Selecta for the kind invitation. Just a brief introduction on the topic. We know that the vast majority of the human genome is transcribed, and the largest part of the transcriptome, of the human transcriptome, is non-coding. And with the non-coding RNA that can be roughly divided in small non-coding RNA, including but not limited to microRNAs, and also a long non-coding RNA that are an heterogeneous class of non-coding RNA that are basically defined as having a length longer than 200 nucleotides and having not a protein coding potential. We know that long non-coding RNA can basically exert function by interacting with proteins, interacting with DNA, interacting also with other RNA molecules, and very often mediated the interaction between these molecules. And in this way, they can control virtually any biologic process, from the gene expression to post-transcriptional regulation and so on. And so it is not surprising with this plethora of function, they can control all biological aspects, including the malignant transformation. So among the cancer where non-coding lncRNA have been described with oncogenic or tumor suppressive function, there is of course multiple myeloma, that is the second most common hematologic cancer, stems from the malignant transformation of plasma cells, and unfortunately still characterized by a poor clinical outcome. And in the last decade, why myeloma is a good model to study non-coding RNA? It is because in the last decade, we and others have demonstrated that the lncRNA landscape is profoundly dysregulated in cancer cells from myeloma patients, and the expression of lncRNA can also predict the clinical outcome of myeloma patients. And also we and others have provided many demonstrations that the modulation of the long non-coding RNA can significantly impact the myeloma cell pathobiology, and also provided the basis for a first-in-human clinical trial using inhibitors of non-coding RNAs. So this being said, my commitment in the lab is to decode the oncogenic lncRNA networks in multiple myeloma. And to this end, we have developed an experimental platform that spans from the identification of functional lncRNA using different screening approaches, then to characterize the mechanism of action of the relevant lncRNA, and finally, we are a translational lab to optimize strategies for the therapeutic targeting of oncogenic lncRNA for translation to clinical trials. So starting from the identification of functional lncRNA, the question is why today, in an era where the genetic dependency of cancer cells has been extensively characterized using different approaches, why in this era we are still missing this information for lncRNA? The reason is that the loss of function approaches like RNA interference or CRISPR knockout that are commonly used to study protein coding genes are not suitable, or at least are not optimal to study long non-coding RNA. And the reason is, for instance, if we look at the RNA interference, is that most lncRNA are retained and exert function in the nucleus, while the RNA interference machinery is mainly cytosolic. Or in the case of CRISPR knockout, while it can work even in high throughput screen, especially using paired guide RNAs, when using single guide RNA, there is the need of a priori knowledge of the functional domain of lncRNA, and this information is still missing for most of them. So to overcome this issue, what we have done is to apply and optimize two kinds of technology to study lncRNA that are, from one side, Cas9 derivative technologies like CRISPRi or CRISPR activation, CRISPRa, that allow the to modulate, in the case of CRISPRi, to down-modulate, in the case of CRISPRi, to upregulate the expression of endogenous lncRNA, or most recently using Cas13 derivative technologies that allow to directly target for degradation the RNA molecules. For today, I will focus on our earlier studies using the CRISPRi. So again, CRISPRi is basically make use of a dead Cas9 KRAB fusion protein, where the dead Cas9 is a catalytically inactive Cas9 and KRAB is an epigenetic modulator. This fusion protein can be guided by a single guide RNA downstream of the transcription start site of gene of interest, including lncRNA. And in this way, the expression of the lncRNA can be repressed, resulting both from a stoichiometric block operated by Cas9 and also an epigenetic modulation operated by KRAB. So in our approach, what we have done to characterize the functional lncRNA in multiple myeloma is first to analyze RNA sequencing data from myeloma cells from patients to identify lncRNA that are expressed in our cellular models, because lncRNA, the expression of lncRNA is highly tissue specific. Then to use this candidate to design and generate pool library of guide RNA that have been used in a primary and then in a validation screen to identify those lncRNA that were targeted by depleted guide RNA that most likely exert oncogenic function in our cellular model. This approach led us to identify at least 40 lncRNA, functional lncRNA that exert oncogenic function and support the proliferation of myeloma cells that we call long DEP, long dependency. And now at this point starts the fun, because our goal is to characterize their functional role, and we are following different directions to prioritize the most relevant lncRNA. For instance, we integrate our screening data with the clinical data in our patients to identify those long depth that can impact also the clinical outcome of patients. Of course, we are also interested in those lncRNA that can have a role in specific myeloma subgroups, or even better, we also have identified lncRNA that are specifically expressed by myeloma cells or plasma cells, and so therefore can represent ideal target for therapeutic approaches. We are also working on we have coupled also our screening data with extensive subcellular RNA sequencing that allow to identify where the lncRNA is locating at subcellular level, so that can indicate some specific function and impact on specific biological processes. And last but not least, in collaboration with Dr. Matthew Disney at the Script Research, we are also trying to prioritize those lncRNA that have druggable structure using small molecule-like compounds. So for today, I will focus and I will discuss our the work we have done to characterize the molecular function of miR-17HG that resulted in our screening as the leading dependency in myeloma, and that remarkably had a dependency score that was on a par with the positive controls included in the screen, like the oncogenic factor c-MYC and IRF4. Of course, we first revalidated the screening data using both a CRISPRi approach in different myeloma cell lines that were infected with inducible guide RNA, and also using antisense oligonucleotides in a large panel of myeloma cell lines that confirmed that miR-17HG is a strong dependency in our setting. So what do we know about this gene? This gene is a known oncogenic factor driver, and it is mainly known to provide precursor for a cluster of microRNA called miR-1792 that we have previously reported with a role in multiple myeloma. But what is even more interesting is that by a deep RNA sequencing analysis, we have found that alternative splicing of this gene can also produce a novel lncRNA that we call RROL, for reasons that I will explain later on this presentation, and that did not have any correlation at expression level with the cluster, with the microRNA cluster, indicating that it can possibly exert function independently of the microRNA. An hypothesis that we indeed validated by showing that depletion of this lncRNA, the effect of depletion of this lncRNA on the survival of myeloma cells cannot be rescued by overexpressing the primer or by knocking out DROSHA, that is the limiting enzyme in the biogenesis of microRNA. Therefore, indicating that this lncRNA is a determinant of dependency in myeloma cells. To characterize its molecular mechanism, we started from two observations. First, that this lncRNA is nuclear retained, and secondly, that downregulation of this link affects the MYC transcriptional activity, suggesting that this lncRNA can cooperate with the transcription factor c-MYC in tumorigenesis. And this hypothesis was further supported by our other finding that RROL and MYC can physically interact in myeloma cells, as shown both in vitro using an RNA protein pull down and an RNA immunoprecipitation assay, and also in live cells by adapting the RNA Hist3 hybrid to study lncRNA protein interactions. Next, by coupling transcriptomic and epigenetic study, we identified an RROL MYC co-regulated gene network controlled by both these factors. And among these co-regulated genes, we decided to focus further investigation and also to use this gene as a bait to study how RROL MYC can cooperate in transcriptional control, the gene ACC1. This because this gene was the top target of both RROL and MYC, and also was a strong vulnerability in myeloma cells, both in vitro and in vivo, as shown in this plot. So to understand how MYC and RROL cooperate in this transcriptional regulation, we firstly demonstrated that RROL can actually interact with chromatin, and this interaction can be detected at a ACC1 promoter. And then we demonstrated that at the ACC1 promoter, RROL and MYC can co-localize, as shown by coupling RNA fish with immunofluorescence. As you can see here, the RROL RNA and MYC protein could be co-detected at the ACC1 gene locus, and also more importantly, by performing a MYC chip qPCR in presence or absence of RROL depletion, we demonstrated that RROL is necessary for MYC occupancy at this locus. So we know that the transcription factor MYC does not regulate gene expression by working alone, but indeed MYC cooperates with many transcriptional co-regulator and transcription factor to regulate gene expression. So our question was, does RROL also mediate protein-protein interaction, so MYC interaction with other transcriptional co-regulator? And to address this question, we analyzed the MYC protein interacting network in two different systems, in an in vivo model using a proximity-dependent biotin identification assay, and in myeloma cells using a co-IP mass spectrometry approach. And in both cases, the investigation of the MYC protein interacting network was performed in presence or absence of RROL depletion. And this analysis led us to identify different RROL-dependent MYC interactors, and on top of them, this protein called WDR82. So WDR82 is a key component of the SET1A methyltransferase complex, which is an epigenetic complex that controls the gene expression by promoting the deposition of H3K4 trimethylation at the active loci. What we demonstrated here about WDR82, and it is summarized in this cartoon, is that RROL, firstly, that RROL can physically and directly interact with the WDR82, and that also it mediates the occupancy of this epigenetic factor at the CC1 promoter, and also the deposition of the H3K4 trimethylation. Overall, indicating a molecular scenario whereby RROL binds to the chromatin, and at the chromatin level provides a molecular scaffold for the interaction of MYC and WDR82 that leads to the transcriptional and epigenetic activation of ACC1. So finally, from a functional standpoint, we know that ACC1 provides encodes for the rate-limiting enzyme in the de novo lipogenesis pathway, that is a metabolic pathway that is known to promote the tumorigenesis. So our question was, does RROL and the MYC-WDR82 complex regulate this metabolic pathway through ACC1? And indeed, we were able to demonstrate this hypothesis, showing that the depletion of RROL, both in myeloma cell lines and in patient-derived cells, reduced the de novo lipogenesis, as shown here by the incorporation of a radiolabeled glucose into the lipid pool, that is a marker of de novo lipogenesis, and also in an independent experiment using a lipid profiling after RROL depletion that showed a specific reduction of saturated and monounsaturated fatty acid that are produced by de novo lipogenesis. Further supporting the role of this metabolic pathway in the RROL mechanism of action, we also found that the supplementation of palmitate, that is the main downstream product of ACC1 activity, can rescue the apoptosis induced by RROL depletion. And so on this base, we decide to name this new lncRNA RNA Regulator of Lipogenesis, or RROL. To sum up this, what we have done to uncover the molecular mechanism of this gene, our proposed model is that the Mir70HG that is under the transcriptional control of c-MYC can be alternative spliced to produce a microRNA cluster or a novel lncRNA. Based on our previous work, we know that the microRNA cluster forms homeostatic feed-forward loops with the MYC transcriptional network to maintain the pro-apoptotic genes regulated by MYC below a threshold level, while via the lncRNA RROL forms positive feed-forward loops that allow and increase the transcriptional activity of c-MYC, specifically driving the activation of the lipogenic program that is essential for myeloma cell proliferation. So finally, that is, we are a translational lab, as I already said. Our goal is to translate our findings into novel therapeutic approaches for translation to clinical trials. We have also done extensive work to develop novel strategies to target RROL and other lncRNA. This is our most recent effort, where we have screened more than 80 different antisense oligonucleotides that had two different configurations, able either to block and antagonize the function of RROL or induce its degradation in RNase H-mediated manner. And to increase, of course, for the therapeutic purpose and to increase the PK profile, the molecules at the end were also connected with a tocopherol domain that allows and increase the cellular uptake. Both inhibitors showed a very strong anti-myeloma activity in 3 different animal models shown in this slide. Firstly, we tested both inhibitor in a plasmacytomac xenograft. where we were able to demonstrate a potent anti-tumor activity, here shown in this slide, and also an efficient uptake of the inhibitor that resulted in a downregulation of lncRNA in tumor cells. Then in a disseminated, a very aggressive model of multiple myeloma, where the tumor growth can be followed by imaging. And most importantly, also in a patient-derived xenograft, where one of the two molecules, the gapmer molecule, could induce the regression of the tumor and work very similarly to our positive control bortezomib, that is the mainstay in multiple myeloma treatment. So to conclude, our take-home message is that in focusing on multiple myeloma, we can of course say that there is a lncRNA addiction in multiple myeloma, and that all the screening, the CRISPRi, but also the other CRISPR derivative technology are very useful tool to map this dependency, and that lncRNA can exert function, of course, in many different ways. But what we have demonstrated so far is that a main mechanism is, of course, the interaction with oncogenic proteins, and this way driving the functional activity. And also that lncRNA therapeutics is something that is real and will become even more important in the future, as the lncRNA is now clear that can be targeted using both antisense oligonucleotide, or more recently using small molecule-like compounds, and this is something we are working now at the moment. And I'd like to thank all the people that have been working on this project and collaborating on this project from many different labs, and our clinical team, and of course all the patients, and also special thanks to all my funding agents, including the American Society of Hematology, the International Myeloma Society, the International Myeloma Foundation, the Miracles from Myeloma Foundation, and the Dana-Farber Sport in multiple myeloma. And thanks to everyone for the attention.
Wonderful. Thank you so much, Eugenio, for that fantastic presentation. So audience members, that does bring us to our Q&A portion. So you can still send in questions right now for both Paul and Eugenio using the questions tab in your control panel. So we've already received some questions, so I'll start off with those. And our first question is about the gRNA design. Which tool did you use for the design? How were the transcription start sites identified?
Okay, this is a good question, and I mean, it's for the guide RNA design, we use the web portal at the Broad Institute, that is a friendly user, where it's possible either to input the ensemble ID of the linker of the gene of interest, or as many times happen with lncRNA that do not have an ensemble ID yet, there is a syntax to input the transcription start site of interest. And for the identification of transcription start site, we had to rely on RNA sequencing, and of course, a future direction is to implement our approach with some cage assay.
Great, thank you so much, Eugenio. We have another great question from an audience member, and that question is the lncRNA CRISPRi library suitable for analyzing other types of cancer cells, or is the library designed tumor-specific?
Yeah, that is also a good question. And of course, before designing the library, we checked if in already available libraries there was a good overlap with lncRNA expressed in myeloma, but unfortunately the answer is not. lncRNA are really, the expression is really tissue specific. So my suggestion is to study first the lncRNA landscape in the context of interest, and then to design custom libraries.
Fantastic. And our next question is, how do you know that identified lncRNA dependencies are produced by the lncRNA, the transcript, and not just interactions with the lncRNA gene in the chromatin?
So for this, this is a very good question, because the CRISPRi cannot discriminate these two functions of a lncRNA. What we do is to provide some orthogonal validation using antisense oligonucleotides, because antisense oligonucleotides will only affect the function mediated by the transcripts.
Great, thank you, Eugenio. And another fantastic question from an audience member, and that question is, in the CRISPRi screen, did you observe any correlation between dependency score and the expression level of the lncRNAs?
So we did not. We checked if the lncRNA with highest expression were also the one with the highest dependency score, and this is not the case. Expression does not correlate with dependency.
Great, thank you again. And we have another excellent question from an audience member. And that question is, would you categorize microRNA genes such as MIR17-8G as a specific class of lncRNA, or do you think MIR17-8G is an exception?
So this is a tough question, and I think there are two possible answers. First of all, microRNA gene can be categorized as lncRNA. They can be categorized as lncRNA that produce microRNA. So this is for sure something correct. If the microRNA gene can work also as a lncRNA that has independent function from the microRNA, this is demonstrated for MIR17-HG, but we don't know yet if it is a more general mechanism.
Great.